Dear children, in this module, we are going to learn about the genetic code. The genetic code is a sequence of nucleotides, that is a nitrogen bases, which is present in the mRNA. And this nucleotides in mRNA, it has the information to make the proteins, that is the protein synthesis which is taking place, or in other words, it has information for translation to take place. We know there are four bases, the nitrogen bases, which are present in mRNA. The information to make one amino acid lies in the sequence of three bases. And this sequence of three bases which has or which determines a single amino acid is called as a codon. We can see codons over here. AUG, three bases together. It's a codon or it's a code for one amino acid. The UAC is the second codon for another amino acid. GUC, CGU, ACG, UAC, all these are different codons which has a code for a particular amino acid. Now let us learn about the contribution which was done by a number of scientists with regard to genetic code. The first contribution was by George Gamow. George Gamow, he suggested that there are 20 amino acids. We know that there are 20 amino acids, and for coding 20 amino acids, the code should be made up of 3 nucleotides. Each code should be made up of 3 nucleotides. Thus, we get 64 codons. There are 64 codons. That means 4 raised to 3 is equal to 64. So, George Gamow, he suggested that for coding 20 amino acids, the code should be made up of 3 nucleotides. As a result, we obtain 64 codons. The contribution by Hargobind Khorana was that he developed a chemical method for synthesizing the RNA molecules with a defined combinations of bases. And these combinations of bases were either copolymers or homopolymers. Homopolymers and copolymers, they differ in that the homopolymers are made up of a single type of monomers. Whereas what about copolymers? They are made up of more than one type of monomers. So that is about homopolymers and copolymers. So Hargobind Khorana, he developed a chemical method in synthesizing RNA molecules with defined combinations of bases. Marshall Nirenberg, he developed a cell-free system for protein synthesis. And lastly, Severo Okawo, he proposed that the enzyme polynucleotide phosphorylase is used to polymerize RNA with defined sequence in a template-independent manner. So these scientists, George Gamow, Hargobind Khorana, Marshall Nirenberg, and Severo Okawo, they contributed to the deciphering of the genetic code. Using the genetic code, there are 20 amino acids which are found based on the codons on the mRNAs. These are the 20 amino acids. And this is the genetic code dictionary, the codons for various amino acids. Now let us learn about the salient features of the genetic code. The salient features of the genetic code. The codon is a triplet. It is made up of 3-letter code. So the first salient feature is that it is a triplet. The second feature is that 61 codons code for amino acids. As proposed by George Gamow, there are 64 codons. Of the 64 codons, 61 code for amino acids. Whereas the 3 codons, the rest of the 3 codons, that is UAA, UAG and UGA, they do not code for any type of amino acids, and thus they are called as stop codons. These stop codons are otherwise called as nonsense codons, which are the terminator codons which terminates the process of translation. So AUG, the codon AUG, it is the initiator codon, which initiates the process of translation. And in addition to acting as an initiator, it is having another function. It codes for the amino acid methionine. It codes for the amino acid methionine. In eukaryotes, methionine is the first amino acid and formylmethionine in prokaryotes is the first amino acid. So AUG has two functions. One, it is acting as an initiate codon, and the second function is that it is coding for the amino acid methionine. An amino acid is coded by many codons, and such codons are called as degenerate codons, except for methionine, which is having a single codon, that is AUG, and tryptophan, which is having the codon UGG. Except for AUG, that codes for methionine, and for tryptophan, the other amino acids are coded by more than one codons, and they are called as degenerate codons. The genetic code is commaless, or there is no punctuations between the adjacent codons. So the genetic code or the codon on the mRNA is read in a contiguous fashion or in a continuous manner. The genetic code is universal. For example, from bacteria to humans, UUU, the codon UUU codes for phenylalanine. Some exceptions are found in mitochondrial codons and in some protozoans. Other than this mitochondrial codons and protozoans, from bacteria to humans, they are universal. The genetic code is non-overlapping. They do not overlap over each other. And the genetic code is unambiguous and specific. One codon specify only for one amino acid. So there is no overlapping, there is no punctuations, and they are highly specific. One codon codes for one specific amino acid. So dear children, in this module we learned about the genetic code. The genetic code is the code which connects together the nitrogen bases or the nucleotides which is present on the mRNA with the sequence of amino acids on the polypeptide chain. Salient features of the genetic code are that it is a triplet code. It is a triplet code. There are 3 stop codons and there is an initiator codon AUG which also codes for amino acid, and codons are degenerate. They are commaless or they have no punctuations. They do not overlap. They are highly universal and unambiguous or specific. Thank you.